Sorry, I'm not feeling very well today, so I would prefer play my recorded presentation if that's okay.
Okay. Title of her talk is Aquaphotomics for revealing the interaction between water molecule and surface, potential applications to predict cell response and biofilm formation. Dr. Xu, please.
Hi, everyone. My name is Junli Xu. I'm a postdoc researcher from University College Dublin, Ireland. Today, I'm going to talk about how to use FTIR spectral to reveal the interaction between water molecule and the surface, and then use this information to predict cell response and the biofilm formation. As Eva has introduced before, the simple definition for biomaterial is that any material used in the medical device and designed to supplement, store, and come into contact with living biological cells. So biomaterials are being increasingly used in the development of new medical devices. There are a wide range of varieties of biomaterials available. Polymeric biomaterials such as nylon and polyesters are widely used because they are biocompatible and easy to fabricate. The common applications include contact lenses and artificial hearts. Metals such as stainless steel are often used as well. They have good strength and excellent fatigue resistance. The common applications include load-bearing implants such as bone plates and pins. Ceramics such as the hydroxyapatite are also frequently used in dental implants and medical sensors, and they are also biocompatible and have good strength and stiffness. To design a desired biomaterial, many factors have to be considered. Since the biomaterial is designed to contact with a biological system, it is of great significance that it shows good biocompatibility, which means the material do not produce toxic response when exposed to biological fluids. The interaction between cell and the biomaterial is essential for different cellular response, such as adhesion, proliferation, and differentiation. So in addition to this, a good biomaterial should have good mechanical properties, such as good strength, good physical and chemical properties, such as high corrosion resistance. The interaction between cell and the biomaterial surface is very complicated. Cell attachment is always regarded as an important step because it is related to stimulate signals that regulate cell differentiation, cell cycle, cell migration, and cell survival. Therefore, cell attachment or cell adhesion on the surface is crucial consideration in biomaterial design and development. As we see from this figure, the first step in the cell attachment or cell adhesion is the interaction between the water and the surface. Due to the surface characteristics, water forms different hydrogen bonding networks at the interface. Then it comes the proteins. Protein is seated on top of the water layer, and finally, cells will approach and settle on the surface of the biomaterial. Based on the surface feature of biomaterial, cells could be happily or unhappily attached, which will have consequence on the subsequent cell response, such as cell spreading, migration.
We lost the sound. June, Lily,
Sorry, that's my real voice now, but I don't know what happened. It should be okay, but maybe I can continue. It's fine. I don't know. That's weird because, yeah, like when I sent you the slide, like the video, I played all of them. Like it's okay, but I don't know what happened. Like, yeah, yeah, I can start it if you want. Maybe I can start share my screen. It's fine. I don't know what happened.
Okay.
maybe you can try to offload and upload again. I don't know what might be the reason.
It's okay. I can maybe talk now by
Yes,
myself.
please. Yes, okay.
Yeah, it's fine. Yeah, it's just like I'm not feeling very well so recently, but I think I can keep on that. Okay, maybe stop playing and I can share my screen.
Lawrence.
Can I start now? Can I? Yeah, but I cannot share the screen. Is this...
Good, we see it.
All right, just give me a second. Is this the right screen? Maybe not. I think. Yeah, I think. Sure. Can I see the screen? Is that full screen? Is it?
Yeah, that's full screen, Lily. Yeah.
Oh, all right. Perfect. Sorry. All right. Sorry about all these technical issues. And yeah, so there are 4 factors that could influence the cell response. And the first one is the surface topography. That is the roughness or any surface characteristics of the surface. And the second will be the surface charge. And this is also regarded as a very important factor. And then the next will be the surface chemistry, which means the functional groups of these biomaterials. And the next one will be the wetability, which means as the degree of the hydrophobicity. All right, and there are some previous researchers shows that different functional groups will produce different cell response. And this research shows that if the surface with the amine group or the couple cell groups, the cells will be very happy and they grow a lot. But if it's a hydroxyl and also the methyl group, the cell doesn't really like it. So there are less cells can be found on the surface. And the next important factor is wetability, which it means the hydrophilicity or hydrophobicity of the surface. So when the water drop deposited on the surface, it will form different shape depending on how wetability or how favorable the surface is to this liquid. And then normally we say if the water content angle higher than the 90 degree, it is unfavorable or very unfavorable surface to this liquid. And then depending on the hydrophilicity, the cells, the water network forms on top of the surface will be different. They will form different hydrogen bonding network. And regarding the cell response, there are different research have been conducted and there is no consensus on how the wetability influences the cell response. Some studies show that the hydrophilic properties of polymers leads to the increased cell spreading and adhesion. But other studies reported that cells adhered and proliferate at the highest rate when cultured on a substrate with hydrophobic surface or at intermediate contact angles. So in this study, we want to see if use the FTIR spectroscopy to predict the cell response, because we know FTIR can contain rich information on the surface chemistry or material chemistry. And we also want to see how can we review the cell, the water and the surface interactions in the FTIR and use this information to predict the cell response. And this is the experiment design. We have 11 materials, and actually it's just 10 biomaterials, and we use glass as a control, which makes up to 11 in total. And then we have the biomaterials. So the first step is to collect the ATR, FTIR spectra. And then we have to grow cells on the surface of the biomaterials. And in this process, we have faced many challenges. For example, some polymers are just too light, and then they keep floating in the culture medium. So we couldn't really like seed the cells on top of the surface. Oh, sorry. And then we have to assess the cell response by using different dyes. And this is the cell characteristics. So the final step is we want to use FTIR spectra and use the multivariate modeling to predict the cell characteristics. And as we talked before, we know like on different surface, the water hydrogen bonding network is different because of the water surface interaction. So we want to use this information, and we want to contain this information in the FTIR spectra. So we use the method developed by a team member. So we use the silicon isolator, and then we press the adhesive side on the surface of the biomaterial. And then in this space, we fill in with water, and sitting about like half an hour. We collect the spectra with the presence of the water. So in this way, we believe that the infrared beams will pass a small amount of the water adsorbed on the surface, and also they will reach to the surface of the biomaterials, and it will contain both the information of the water surface interaction and also the chemistry information of the biomaterial. And this is the ATR FTIR spectra in the dry and wet state. So in the wet state means we collect the spectra with the presence of the water. And as we see from the spectra, the wet spectra in blue color, it shows all the features in the dry spectra in a red color. And in addition to this, the wet spectra also presents some OH groups in the 3,600 to 3,000 with numbers due to a small amount of the water molecules present at the interface. And we can also tell the OH band shape and also the area is different based on the different surface. And this is because different interactions occurred at this interface. And we see on the glass, the OH band is really broad, and the area is the largest among all these materials. And the PTFE and the silicon rubber, they presented the smallest area and the smallest band. And then we have the cells stained with different colors. On the merged cell image, we can see the green color has been used to stain the actin cytoskeleton, and the blue color is to stain the nucleus, and the red color, which presents different like red dots, and this is to represent the focal adhesions. And we have to apply the manual segmentations on each image. So on each channel, we have to build a mask to highlight different areas So for example, for the cytoskeleton, we have to highlight the area of a cytoskeleton by the masking. And then based on the mask, we can calculate different parameters, such as the area of the spreading area, which means the cell area. And then we can calculate the cell length and also the cell width. And the aspect ratio means dividing the cell length by the cell width. And for the nuclears, we calculated the area of the nuclear. And we also compute the circularity. And for the focal adhesions, we built many masks. And we calculated numbers of the focal adhesions on each cell, and also the average size of each focal adhesion, and the total area of the focal adhesion. And lastly, the percentage of the focal adhesion area to the total spreading cell area. And this is for representative images we selected for each surfaces about how cells grow on them. And we can see on different surfaces, cells present different shapes. And so for example, if we look at the glass, the cell shape is considered to be large, and they have present a kind of relaxing state. And this is because the cells like this surface. And then we can also see there are a lot of the red dots. That means a lot of red focal adhesions on the surface. But if we look at the PTFE, the cells looks quite strange, and they're kind of like elongated and stretched. And this is because they don't like this surface. So when they deal with this unfavorable surface, they are trying to seek for another surface that's better suit them. So you will see they're elongated because they are searching for a better area for them. And also on these unfavorable surfaces, there are less red dots, there are less focal adhesion points. And we calculated the statistics for all these parameters regarding the focal adhesion, and present here. We can see on different surfaces, it shows different characteristics. And then we built the models. In the training set, we have 8 materials. And in the test set, we have 3 different materials. So that means that the samples in the test set hasn't been used in the training set, make them independent. And this table shows the model performance. And on the left, it's the model performance from the dry spectral, and on the right, it's from the white spectral. So in general, we noticed that models built from the wet spectral performed much better than the models built from the dry spectral. And this concludes that the incorporation of the water and the surface interaction will really improve the model performance. And we also noticed the best model is to predict the total area of focal adhesion, and it's using the wet spectral. So it leads to the determination coefficients of the prediction set 0.94, which is quite nice. And then we plotted the measured values and the predicted values of the focal adhesion area per cells, and the result is quite satisfying. So we see all the data points have been distributed closely to the regression line, and even for the prediction side, it's quite nice. And the next, we want to better understand how a cell interact with surface and how this information related to the water and the surface interaction. So we look at the regression vectors. And for this regression vector, we can see the OH stretch has present a very broad band. And this is because the water is stopped on the surface. And then the next is the stage stretching. And we see the highest or the largest contributions coming from the CO double bonds, which is in the region of 1800 to the 1700 wave number. It presents a large positive band and also a very sharp negative band, but both of them have presented the largest amplitude, meaning they have the biggest contribution to the model performance. So based on the regression vector, we can lead to the following conclusion that the contribution to the focal adhesions follows this order. The CO double bond is higher than the OH stretching, is higher than CG stretching, and this result is consistent with the previous research. And we also look at the total area of focal adhesions, and it follows this order. So the glass has the highest total area of focal adhesions, and then it's PVC, PLA, PU, and PET. And the glass has the highest, and this is because glass has a very broad band of the OH stretching, which is a positive in the regression vector. So it leads to the highest focal adhesion. But besides to the glass, the other four polymers, which also has largest focal adhesion, they all have the CO double bonds. And this again confirms that the CO double bond has the biggest contribution to the focal adhesion. But we also noticed that in the CO double bond, they have the positive and the negative peaks. So we want to understand why there is a positive and a negative peaks. So to understand this, we have the difference spectra, which is obtained by subtracting the dry spectra from the wet spectra. So in this case, what's left of is only the water and the surface interaction. And we look at the focal adhesion the orders again. That's PVC higher than POA and higher than PU and PET, which all have the CO double bonds. And then we look at a different spectra. So if the water and the surface interaction is very strong, it will present a very negative peak around the 1700 wave number. So the first is PVC, right? PVC shows a very small negative peak, and then POA shows a bit larger negative peak, and then the next is PET here. We can see the negative peak is much stronger, and then the PET, the negative peak is the largest. So this peak is related to how the carbonyl to water interactions in the form of when the one oxygen atoms, it forms hydrogen bonds with the hydrogen atoms in the water molecule. So the larger this negative peak is, the stronger the interaction is. But then we see the extent of the interactions is in the opposite orders with the orders of the focal adhesions. That means when it forms the interaction with the carbonyl to water between the polymer and the water molecules, they will discourage the cell adhesion because they are in the opposite orders. And we also want to know if this is for sure or is this caused maybe by the water wettability. So we listed the contact angle order here. This is PU higher than PVC, higher than PET and the POA. It doesn't really relate it to the focal adhesion orders here. Okay. And we have published this work last year. So if you are interested, and you're welcome to have a read, and we can discuss more maybe. And currently we are working on the biofilm formation prediction. So biofilm forms when bacteria come into contact with the substrate and then begin to release the EPS. And then when the bacteria has been attached, they begin to replicate and increase their EPS production and secure the structure of the surface. So we are using the crystal valley staining to quantify this biofilm formation. And the next step, we will use the ATR FTIR based on the multivariate modeling to predict this quantified biofilm formation. But currently, this study is still in the early stage, so I don't have much more results to show. So that's for the bio. Thank you. Thanks.
Thank you very much, Xu, Dr. Xu. So very systematic research. We can accept 2 questions from first one, the Professor Takeuchi.
Mm-hmm.
Professor Takeuchi, please. Professor Takeuchi, or yes, please.
I just write a comment in the chat. Later, please check it.
Mhm.
I
Okay.
mean, yes, just a comment.
Okay.
Thank you.
Thank you very much for the comment. Professor Hayashi, you have a question about the.
Yes. So
Please.
first of all, thank you for a great talk. And so I'm interested in the order of the cell adhesion. So CO functional group is the most adhering, but I think there is extracellular matrix adsorbed proteins between cell and the biomaterial surface. So if you consider this structure, so why CO functional group is really promote the cell adhesion? This is my question.
Sorry if I understand you correctly. You mean the surface, like the roughness, also affects the cell response? Is that right?
If you put the cells on a biomaterial surface, I think before the adhesion, protein molecules in the cell culturing media may adsorb on the surface. So I
Yes,
think the protein layer is sandwiched between cell
exactly.
and
Yeah,
biomaterial surface. So
yeah.
so if you consider this structure, why CO functional group is most effective to promote the cell adhesion?
Yeah, so yeah, as I showed the slide before, actually it's water reaches the surface first. So water will like forms a layer on the surface, and then it comes the protein. The protein is absorbed, and then the protein is seated on top of the water layer, and finally it's the cells. So yeah, as you said, it's different layer things, but then the surface of the biomaterials, the different characteristics like the surface roughness or the surface charge and the surface wettability, they all influence how the cells will adsorb by different ways. Sometimes by how the water molecule has been adsorbed, sometimes by how they influence the protein adsorption. So it's very a complicated process. So what we want to do is we want to use ATR, FTIR to present as much information as possible and use this information to predict the cell response. And by looking at the regression vector, we see the CO double bonds really influence the cell response because it's related very well. Yeah, thank you.
Yeah, thank you for your answer. Yeah, exciting.
Thank you.
Yeah, very nice talk. So it's